Hi girls, it's Mrs. Vasquez. So here is another video for us to kind of catch up with the material. I definitely wasn't expecting to make a second video this week, but I hope you guys have been enjoying your week off, definitely getting some studying done and preparing for the AP March exam, hopefully. And also, of course, you have power and you're staying warm. It looks like we'll probably be back to school on Friday, but even so, it doesn't really matter too much for us because we won't be meeting anyway until Monday. So there is some more work that we're going to have to do to keep on pace. Okay, so at this point, you should have seen the two earlier videos that I posted. I think it was Monday night I posted them on the evidence of evolution. That is going to finish our previous chapter, our chapter on Darwin and natural selection, our introduction to evolution. With that, you had to take notes, so I'll be checking those notes as well as the notes on this video when we meet on Monday. And then the second assignment I had posted and emailed you guys about was a video on the biology behind skin color, the evolution of skin color, along with 3 questions. And that sheet I also posted and the video on the website. Alright, so at this point you should have all of that completed and done. This will hopefully be the last assignment you guys will have to do before we come back to school. But it's mostly because we've just missed a lot of time. We need to make sure we finish chapter 1 before we break for the exam and spring break. But chapter 21 will be our final chapter before the March exam, and it's titled The Evolution of Populations. So before we go into the notes, just a heads up that this lecture will be on roughly the first half of the chapter. It's not a very long chapter, but we're going to be covering, or I'll be covering, the beginning of this chapter, close to about the first half of the chapter. And then when we come to class on Monday, after we kind of review and you have a chance to ask any questions on any of the homework pieces you've been doing this week, we're going to finish up chapter 21, and then most likely on Tuesday, if we are able to finish on Monday, Tuesday we'll start reviewing for the March exam, which will be mostly in the form of doing FRQs on review topics. Okay, so chapter 21 is titled The Evolution of Populations. It's our second evolution chapter, and in this chapter we're going to go into a lot more specifics as to how evolution works. We've talked about, we talked about it in the previous chapter, but more so in terms of overall. Like, yes, natural selection is the mechanism behind evolution, but how does natural selection actually cause change in evolution? And so chapter 21 will focus a lot on the genetics of populations, so how gene frequencies and allele frequencies change from generation to generation. The second part of this, so population, just keep in mind that when we were talking about natural selection and Darwin's observations, we tended to focus a lot on the individual because that's what natural selection acts on. It acts on the individual. So actually I'm going to move ahead to this first bullet. When we talk about natural selection, you're looking at, is the individual, does it have an advantage? Is it able to camouflage better or be faster or smarter, do something that can help it survive? So we talk about individuals, but remember that individuals themselves, we don't evolve alone. It's populations that evolve. So if you have enough individuals that are being selected on by nature, is that going to cause enough of a change in the gene frequency that will eventually cause an entire population to evolve? And so in the previous chapter, we tended to focus a little bit more on the individual, but in this chapter, we're going to focus more on population. So just a reminder, hopefully, for those of you who have been reviewing ecology for the March exam, we'll hopefully remember what a population is. And a population is a group of organisms of the same species that live in a particular area together. So you can have a population of bald eagles in New York State, or the population of humans in Rye. Or you can have a population of humans in the United States or the Eastern Coast, whatever your region or location is. Okay, so evolution is genetic change, but specifically it's genetic change occurring in populations. You can talk about evolution in the entire species as a whole, but we're going to be talking about evolution on the small scale, population scale. This small scale is also known as microevolution, micro being small. I just want to kind of show you this picture over here first. So before you write down these notes, this picture over here I think is a great picture because it kind of shows you the two major ways of looking at evolution, the 2 types of evolution. We're going to be focusing on the right. So evolution of particular populations. These are small changes, changes you can see from generation to generation, and those small changes are reflective of changes in the gene pool. So the gene pool is all the alleles, all the genes of a given population, and so basically what it means is, okay, say you have a population of deer and they have a high diverse gene pool, that means there's a lot of variety in the alleles and a lot of different types of genes. There might be a lot of individuals of deer and they are just representative of a lot of different phenotypes and alleles and variety. So the more diverse a gene pool, the healthier that population is. But if you have a population, for example, maybe it's endangered, or maybe it's just a very small population that's isolated, well, the less individuals you have, the less opportunity you have to have diversity, the less alleles being represented, the less phenotypes as a result being represented. And usually this means not a good thing. So you have a less healthy species. But just to go back to microevolution, microevolution is genetic changes within populations. This is different from macroevolution. So macroevolution is like big scale changes. Think, amphibians evolving from fish, and then reptiles evolving from amphibians, or humans evolving from earlier smaller primates. Those are big changes that happen over millions of years. That's probably what most people think about when they think of evolution, but we're going to be looking at smaller changes, microevolution. Okay, and so here's an example of microevolution. This particular finch, so go back to the Galapagos Islands in Darwin. On one of the islands, remember every island had kind of their own species or subspecies of finches. And I forget their last name, but Rosemary and Peter, this famous couple, the scientist couple, I'll get you their names, their last name on Monday. They've gone back to the Galapagos Islands almost every single year for the last 50 years. They're still alive. They're really old right now, but they've done they've kind of repeated a lot of the observations that Darwin made and experiments, and they have taken so much data from the different species on the Galapagos Islands, particularly finches. And what they noticed, one of the things they noticed was pretty remarkable. They actually witnessed and observed microevolution happen just over a few years. And so what happened is on this island in 1977, so in between these two bars, there was a pretty significant drought on the island. And after the drought, there was such a big drop in population size. I think they had about 2,000 finches maybe documented before the drought, and after the drought there was only about 300 that were found. And so this couple, Rosemary and Peter, what they did was they took as many of those 300 finches that they could, and they were trying to figure out what about these 300 finches, which was such a small percentage of the original population, what allowed them to survive? What caused them to survive over others? And they took lots of different types of data. But what they realized is that when they took the beak length or beak depth of these finches, and they took the average. So look over here. So the average of the finches that survived after the drought, look at how much higher it is. It's about, 9.7 maybe? And then they compared it to data they had took years earlier before the drought, where the average of beak size or beak depth was about 8.9, let's say. So not a huge difference. I mean, from the naked eye, you probably can't notice a 1.5 millimeter difference. But when you actually record the data and put it into charts and document it, you start noticing these differences a little bit more. And so what did they notice? Well, something about the drought caused finches with longer beaks to have more success and to survive over those that might have had the original shorter beaks. So why is that? Well, remember, if there's a drought, the surface of the soil is the driest, right? And the deeper in the soil you go, the more moist you get. So maybe the finches that had the longer beaks, maybe they were able to reach deeper into the ground and catch more insects who were digging deeper into the ground to try to get more moisture. And so why is this an example of microevolution? Well, not only were the surviving finches the ones that had the longer beaks, but the finches in the generations after them also maintained that longer beak. And so there was sexual selection. finches kind of saw that there was this advantage to having longer beaks in times of drought, and so they were selecting for finches that also had those longer beaks. And so we notice a pretty big statistically significant difference. And so the genotype frequency, the allele frequency changed over just a couple generations, not even. Okay, so this is a pretty classic example of microevolution, but what contributes this genetic change? So we need some sort of genetic change from generation to generation in order for evolution to happen. Well, a lot of these things that contribute to it, we're not going to touch on all of them right now because that will be the end of the chapter, but just to highlight a few things, mutations. So remember point mutations, frameshift mutations, chromosomal issues like deletions and duplications, translocations. And then, of course, sexual reproduction and meiosis. So remember, one of the most significant differences between meiosis and mitosis is that, and as a result, sexual reproduction and asexual reproduction, is that sexual reproduction leads to so much genetic variation. You have total recombination of the chromosomes and the alleles. You have every single egg cell is practically different and unique from other egg cells, and same with sperm cells. So it starts with crossing over. So remember, in prophase 1, we have homologous chromosomes lining up, and to make tetrads, remember homologous chromosomes are, matching chromosomes. So the one you inherited from mom pairs up with the one you inherited from dad, and their genes are swapped. That's what crossing over is. Something else was independent assortment. So in metaphase, when the tetrads are lining up in the middle, well, the direction in which they line up and the order, order in which they line up, that's completely random too. And when that egg and the sperm, which all of them are so different from each other, every sperm that fertilizes with a random egg, well, every offspring they create is all going to be different. And it's so random. Whatever sperm is fertilized with whatever egg, is not done intentionally, is done randomly. Okay. Oh, one thing before I move on to these two pictures over here, we have Darwin and Mendel. Why these two particular? We haven't talked about Mendel in a while, but why Mendel? Because this chapter is kind of bridging together this idea of genetics and evolution. So Darwin and Mendel were alive around the same time, but they never really knew each other or knew of each other's work and research. It only was until years later that we realized that evolution and genetics are not two separate topics. They actually are the reason for each other, especially for evolution. It explains how evolution happens because the genetics changes. The genetics differs from generation to generation. Okay, so this leads us to our next topic, which will be our main topic for the rest of this lecture, is this idea of being able to calculate genetic frequencies. So can you figure out the percentage of alleles? So dominant and recessive. Can you figure out, okay, if there's, out of a population of 1,000, 9 are short versus the 991 that are tall. Can you figure out the percentage of heterozygotes or homozygous dominant organisms? So right now that might seem, well, how the heck would I figure that out? Well, this we'll be talking about now. And it's based on this idea that was developed, this principle that was developed by two, one was a mathematician and one was a scientist, Hardy and Weinberg. And so Hardy was a British mathematician, Godfrey Hardy, and Wilhelm Weinberg was a German physician. So early 1900s, specifically 1908, these two, I guess a mathematician is kind of like a scientist. These two scientists, they were studying evolution and microevolution specifically, and populations and genetic frequencies. And they came up with this idea or this theory that, okay, populations, yes, they evolve. But if a population is not evolving, that means it must not be experiencing genetic change, because genetic change is necessary for evolution. So they said, hypothetically, if a population were not to evolve and not experience any genetic change, what would be the conditions that would have to be in place for them not to experience this? And so the first thing they said was no mutations. So if a population is not evolving, that must mean there can't be any mutations happening. Because remember, mutations lead to change. And so if a population is not experiencing evolution or genetic change, It cannot have any mutations. That's number one. They also said there has to be random mating. So random mating, remember, so I'll actually bring up number 3. So random mating is pretty much the opposite of natural selection. It's pretty much the opposite. So unless you are in an arranged marriage, everyone is intentionally mating, right? And you pick a mate based on maybe physicality or personality or intelligence, whatever matters to you when you're looking for a significant other. You pick someone based on traits, right? You're selecting for a particular person. That's not random mating. Random mating would be you just randomly pick someone, you mate with them, you have, relationship or not with them, and then you move on. You're not picking someone based on certain characteristics, because the idea is that if you are picking someone based on certain characteristics, which pretty much everyone does, you're selecting for certain traits, and over time that could lead to change. Okay, so 2 and 3 are very similar. Number 4 is extremely large population size. So the larger the population, the more stable the population. And the more stable, well, the less likely that any tiny changes could have any long-lasting impact. And then finally, the 5th one is no gene flow. So gene flow we'll talk about a little bit more at the end of the chapter, but think of gene flow as migration. So gene flow is the movement or the migration of any genes or phenotypes or characteristics, traits that might be going into a new area and introducing new genes, new alleles into a new species gene pool. Okay, so think of like invasive species is a great example. So invasive species definitely changes the gene pool, adds new alleles. So that would not be contributing to a population that had no evolution. Okay, so what Hardy and Weinberg said was that if these 5 things are happening, that means that population must not be changing. So the percentage of any alleles that exist in their gene pool must be exactly the same from one generation to the next. But if you're reading this and you're thinking right now, well, what's the point? Because I can't imagine any population fulfilling all 5 of these conditions at the same time. You're right. It's kind of hypothetical because in real life this really doesn't exist. So why is this important? Well, knowing this led to Hardy and Weinberg coming up with a couple important equations, because if this could be true hypothetically, it could kind of set the stage for being able to determine allele frequencies and genotype frequencies. So I'll explain a little bit further. Okay, so if at least one of these conditions is not met, then the population is evolving. So this was a conclusion that they were able to form, and people today can still form, if you can determine that at least one of these things is happening. Okay, and this is known as the Hardy-Weinberg equilibrium, sometimes also known as the Hardy-Weinberg principle. Obviously, it came into be because of these 2 people, Hardy and Weinberg. Okay, oh, and here's them. Okay, why is this really important for us? Because knowing this principle helps us use the following 2 equations that you're going to be learning about that are associated with this principle. And it helps us to calculate the frequencies of alleles in the population to prove that evolution is happening. So, for example, if you have a gene pool and say out of all the alleles for, I don't know, let's say there was brown hair and blonde hair. Obviously hair color is more complicated than that, but let's just say, there was 2 alleles. Capital B was for brown hair, lowercase b was for blonde hair, and everyone in a particular population had a combination of these 2 alleles. If, say, the brown hair was occurring in the gene pool 60% of the time, that means out of the entire population, 60% of those alleles were going to be brown, capital B, which means 40% would be blonde, the other allele. So we can actually, once we kind of know some information, we can use information, particularly in word problems, which you're going to be doing in the packet that I'm also posting. You can actually determine the percentage of different alleles, the dominant and the recessive, and then you can use those to actually use a second equation to figure out what percentage of the population is heterozygous, what population is homozygous dominant, what percentage is homozygous recessive. And this is all part of what we call the Hardy-Weinberg principle. And based on this idea that if evolution is not happening, these conditions can be met, and these conditions lead to these equations. Okay, so I'm not going to go into too much detail about these equations yet because the second video you guys are going to be watching is going to be the Bozeman video. So Mr. Anderson is going to go into more detail about this. I also don't want to make this video super long, so I'm going to kind of end it here, but I just want to show you really quickly is that there's two major equations. The first, all of them have to do with two letters. These are the two big letters, P and Q. They're variables, but they're specific variables. So P is always going to be representing the dominant allele, and Q is always going to be representing the recessive allele. Okay, so in its frequency, so percentages. Because you assume there's only two alleles for every given gene, both of these two alleles have to add up to 100% or 1. Okay, these alleles are sometimes given as a percentage, especially when you're talking about frequency, but they're also written as decimals. And so you're going to have to convert, we're going to convert decimals to percentages depending on what the question is asking. So just be prepared for that. Okay, the first equation you're going to be looking at is P plus Q equals 1. So the amount of P's and the amount of Q's, so the amount of dominant alleles and the amount of recessive alleles have to add up to 100%, right? Because these are the only two that exist. So whatever amount you have of one, the remainder to 100% will be of the recessive. Okay, and then I'll just note these are some modifications you can make depending on what the question is asking. You can use it to your advantage to reword. Okay, I'm going to stop here. The Bozeman video is going to go over a second equation. This one, so the first one talks about allele frequencies. The second equation is going to look at genotypic frequencies. So you're also going to have P's and Q's, but now P squared is going to represent homozygous dominant. So not just an allele, but a genotype. 2pq is going to be the equation or represent the heterozygous genotype. And then q squared, remember q is the recessive representation, q squared is going to be homozygous recessive. So if you can figure out your allelic frequencies, you're going to be able to use that to figure out genotypic frequencies. Okay, so I'm going to stop right here. We're going to go over a little bit more about this on Monday, my side of the notes, but also now watch the Bozeman video. You'll be able to get some practice examples with Mr. Anderson there. And I think he does a really good job of explaining and breaking it down. The beginning of the video kind of is a little bit repetitive. It seems that he doesn't assume that the watcher is going to know like phenotype and alleles and genotype and homozygous dominant. For you guys, that's not new information, but go through it slowly. It'll be a good review anyway for you guys. And as the video goes on, he'll get into specific examples so you can see how it works out. And then the packet that I posted, try to answer as many of those as you can. Okay, if you have questions, that's totally fine. We'll go over it in class. But try to do as many as you can and see how well you can pick out the information. Okay, so thank you guys. This was a little bit longer than I was hoping, so sorry about that. But make sure you have these notes taken down and have as much of that packet as filled out for Monday's class. Okay, see you guys on Monday.